We've just heard three excellent papers on collaborative work. And here's a candidate for some more collaborative work. I'm going to be talking to you about something, as they used to say in Monty Python, and now for something completely different. And the advantage of that is that you can approach, as you know, many targets from many directions. But to our knowledge, I don't know of anyone who's approached the phenomenon, the fact of rapid replication as a property which is a deserving target. And that's what the replicants do. The replicants were first discovered in brain cancer cells growing in tissue culture in the laboratory. The way they were discovered was when the brain cancer cells were rapidly replicating, they produced 5 times as many cells in 1 week time. And the protein that we were looking for, we thought if we're lucky it'll still be there. In fact, it was not produced in 5 times. It was produced 10 times as much. The concentration of that protein was 10 times during rapid replication. Now, that we didn't understand at all for several years, and then began to dawn on us that there was a chemistry which was switched in with rapid replication. We'd all been so absorbed with the DNA that we've learned to work with for 50 years. Nobody apparently has asked, is there a change in chemistry when one puts one's foot on the gas pedal and goes fast forward? And this is the chemistry of rapid replication. Now, replicants are gene peptides. Their sequence is 77 to 50 amino acids long. They're rich in lysine and histidine. And one example is shown above, KAGVAFLHKK. I have a friend who's put that on his sneaker when he's running, and when asked, what is that? He starts on his lecture on replicants. It's simply an example. It's, in fact, the first replicant that was identified in glioblastoma multiforme brain cancer cells. And what replicants are good for so far, as far as we can see, is 2 things. One, they give you a chance at disease surveillance, which searches, identifies, and counts. Replicant count equals number per 100 amino acids in the gene by year, by kingdom, by species, by strain, by gene, by protein, by country, etc. You can ask in the software we have designed any of those questions and get an answer as to the concentration of the replicants or the replicant count in the gene under that particular condition. And so what we found is that it's useful for that. And we have studied now 16 viruses which have had 41 outbreaks. And in all of those, there has been an increase in replicant count which preceded the outbreak and accompanied it, preceded by 1 to 2 years. Now that means that the age-old disadvantage that we have been operating under, that is the annual, constant surprise. Oh dear, here's another outbreak of something or other. The ignorance that's involved in that is centuries, years old. We now have an opportunity to address that ignorance and know 1 to 2 years ahead of time what's coming, what it's going to hit, how hard it will hit, how lethal it will be, and a few other things. Now, here, for example, in the Ebola outbreak of 2014, last year, In March, you remember, the World Health Organization said, we're worried about Ebola. It's not this localized little thing. It seems to be spreading, and pandemonium resulted. So we thought we should have a look. We're a little bit like ambulance chasers. When something comes up, we do apply our search to it. What we found with the first search was annual replicant counts in Ebola virus in all strains in humans and animals, all together. What you see is annually from 1976, the replicant count in blue is the mean count that year, red is the standard deviation of the mean. Now what you'll see is that the standard deviation of the mean and the blue, the mean and the standard deviation both rose significantly in 2013, 1 year before the announced outbreak. Now, if we'd have been doing this, we would have known in 2013 that something was about to happen. We didn't have enough courage to bring that out until about September. And when we brought it out, it had already dropped. So we had to put into our announcement, not only did the replicant count go up in 2013, it's now dropped, and folks, it's all over. Now that takes a certain kind of mental condition, which I think you can understand is anxiety provoking. In the middle of everyone announcing that we're going to have 1.25 million cases in January, we announce it's over. And in January, it was over. It was over. So you can't do that by automatic analysis unless it's so. And if you do it 800,000 plus times sequence analysis, which we did, and it's always the same answer, then there's no little bug in the system. That is a fact. So we went to look at other things like the proteins. Ebola Marburg, there it is again. This time you could see it beginning in 2012. 2012, 2013, and it drops. Another protein. Ebola polymerase, 2012, 2013, then it drops. Another one. Ebola, VP35, another protein. Here in 2012, 2013, and it drops. You realize these are independent searches. All you do is you punch in, you type in, I'd like to know about Ebola VP35, please. And then you go to sleep. If it's a big search, this is not a very big one. This is only 99. You don't have to go to sleep. In about an hour, you have the answer. And the answer is clear. And if you compare it with what we'd known now for several years before, that is that there was a rise in H1N1 preceding the 2009 pandemic. You see, that rise was a gradual rise, not like this one in Ebola. In contrast, here this is the fifth search, the Ebola glycoprotein. Now you have 5 independent searches which show this increase in 2013 and a sudden drop. And in contrast, in Canada in 2008, you see, this is from 1976 also, just to compare the time period with the time period for Ebola. In 1976, you have these values annually. In 2008, it's gone up. And in 2009, you have an outbreak, which is then called a pandemic. And then it drops. But it doesn't drop so dramatically, you notice. It drops halfway. And then it started up again. And folks, you could have known this when we did retrospective examination in Mexico, where it all started. Every year from 2003 to 2009, you had an increase. And if you did several other countries in Japan independently, you got the same increase. So that what we have seen here is a forewarning several years in advance of a slowly rising replicant count. This means, no surprise, these viruses don't just appear suddenly, except as they did in Ebola. They usually have a gradual buildup. Now, we were very worried about that, even at that, even with this demonstration, until we remembered we'd shown the same thing in 2002, 2003 for SARS. And nothing was happening from 1995. See that? The beauty of this method, by the way, this system is you get values that are reproducible year after year, and then you get your change. And there's your change with your standard deviation up there. And it dropped suddenly. And you know that we owe a lot of credit to the public health efforts in SARS. They were much better than they were in Ebola. But it was all over. When SARS started, it was over in 8 months. And the gene told us so. And the gene will tell you so in every one of the tests we've done so far. We have not had anything yet where there was either a false positive or a false negative. I know that's hard to take, but that's the fact. Anyway, on October 9th, we announced that there was this increase. It was published. And I'm sorry. And then we had to announce that the new cases were going to drop also. So on October 9th, a recent sudden drop in the replicant count may depend on early end to the current outbreak. And then we said something about public health measures if they kept up with those viruses which were already in humans. Now, the next step, obviously, was to select our replicants and to see if we could make a synthetic vaccine, as we had successfully done in H5N1 and had also done in Tower syndrome virus in shrimp. And both of those were effective based upon the same principles. You detect the replicant increase, you synthesize by solid phase synthesis. In 7 days, your vaccine, and it's freeze-dried, and you can ship it to the most remote areas without refrigeration, and it works. And here we are. This is where we were with our last results a couple of weeks ago in making a replicant vaccine blocker. It's called a blocker because it has immediate effects on blocking, and it has long-term effects on immunizing, which is the immunogenicity part. And this is Ebola, and you can see that you get a healthy response. And the next step is challenge, of course. And this is now why we're calling for partners. There's too much here for a small group to do, and lots of good things to be done. And so we're calling for partners, and we're forming a replicants vaccine consortium. And we're delighted to hear about the other consortia that are working, and we'd like to follow suit and have the same thing. So anybody who'd like to join us, we're delighted to have you work with us. Just quickly, here's the first genomic prediction and tracking of influenza. This is influenza H1N1, the pandemic of 2009. And here, every couple of days, all the results are not included. But you can see the reds are the infectivity gene, replicant gene, and the blacks are the lethality gene. You remember at the beginning of that pandemic, the mortality rate was much higher, and people said, wonder why it dropped. Remember that? Mexico, beginning, high, gradually down, then over. But not over for infectivity. So everybody says, the pandemic's over, and in 2010 we have another little bit. 2011 we have another one. Well, the gene infectivity level got up to around 9 or 10, the mean level, in Mexico, as it did here. This is overall, worldwide, global. And now the replicant count in Mexico is 13.5. We're in for another one. We're getting another major pandemic of H1N1. And now we have evidence of this in Canada. Over the last 2 years, they had big outbreaks. And now we have evidence in India. where they had high mortality and are wondering whether it's going to go away. So we're on our way to another one predicted by that persistent infectivity gene, replican gene. I'll go very quickly. You can see it. It sounds hard to believe, but as Kekule was justified when they finally could see the six-membered ring, there they are, the replicants crawling on the surface of the hemagglutinin gene in blue. Here you see, oops, sorry. Here you see the blue covering part. This, a lot of it's not covered of this HA. This was in 2002, replicant count 3.2. 2008, replicant count 5.5. You can see the replicants beginning to cover the tip, which is the insertion point for the hemagglutinin gene. And the red is the contact point for sialic acid. And things come around. That was the basis of my thesis 100 years ago. And here it is being encircled by the replicants. And there it's almost completely encircled in 2009 when you get your outbreak. Now, what those replicants are doing, we don't know. Are they helping the injection of the DNA? Are they protecting against the antibody? We haven't a clue. Here's a retrospective going back for each of the major outbreaks of the last century. And each one is accompanied by a rise. I'll have to go very quickly now. Here we have the comparison of H3N2 in global and in global rise here. We have a rise in China. If you ask, well, what's it doing in China? If you ask what's it doing in Ontario, Canada, it's the champ. And so it went, in the last couple of years, building H3N2. Now you realize that we would not have had all the deaths due to H3N2. We realized, oh dear, we didn't include H3N2 in our vaccine last year, and so more children died. We'll try and do better next. Well, if you'd had this data and used this data, it would not have happened that way. And if you don't use it now, it's going to happen again. So, as I said, in each of 41 outbreaks, 16 viruses, 1 million gene sequences. For instance, in Ebola, I said we had 800,000 sequence examinations. In H3N2, we had 528,000. H1N1, we had over 1 million. We now have dengue. And by the way, these vaccines can be used as adjuvants, so that for those nice papers that we just heard, there's no reason not to have what we call a two-punch vaccine added as an adjuvant. It'll work as it's working now. Effective synthetic replicant vaccine is produced in 7 days by solid phase synthesis, freeze-dried, without limitation of number. So we really have a chance to get to the 6 billion people on the planet who are not being served now. Only 1 billion are being served. 6 billion are not being served. Here's just a quick example of how we examine the lethality gene in terms of H5N1 in birds and in humans. And you can see there are 8 genes in influenza, and they don't show much replicant count, but the middle one, the PB1, is the one that's very clear year by year. And so you have an example of how you go at those analyses. Here's another example, replicants in human cases where you have the replicant count, the number of cases, and the mortality rate all going up. And by the way, you see this cycle, which is repeated and repeated and going up. And here it is for localization. You can localize whether the country. We predicted the Indonesian outbreak and the Thailand outbreak when all the other countries weren't doing anything in 2000s. Host localization. You can tell, you see H5N1 likes people, unfortunately, better than likes chickens and ducks. This is just to show that many of these, not all of the replicants, are conserved so that you can look to a pan-influenza vaccine. These are examples in chikungunya, and you can see two examples here. One is here with just straight replicant count going up to a high, and here it is just those replicants above a count of 4. 4 is an arbitrary number. It's chosen on the basis of what we've observed. If your count is below 4, you're usually resting. When it's over, you're not. And dengue. Dengue had its high in 2012, and we're now observing it in increased activity there. And here it is in hand, foot, and mouth disease in China, tracing its increase as the cases went from 50,000 to 1.9 million. Here it is for hantavirus. We haven't found one yet where there is not a correlation between the replicant count increase and the virus. So the ability to obtain advanced replicant gene notice of coming outbreaks and rapidly to produce and test accurate and effective vaccine blockers 1 to 2 years in advance of the outbreak has led to the formation of the NTD Replikins Vaccine Consortium. Additional partners are sought for the consortium to facilitate global surveillance and more effective rapid response. Thank you very much.